Hi everyone, we're going to go into the next set of talks. So this is going to be the data-driven discovery session. We've got three really interesting, exciting speakers coming up, and we'll have a panel discussion afterwards. And this session is really going to be highlighting, how cohort research discoveries can be enabled through big data and data standards and interoperability. So please stay for the session, and I'll get our first speaker to join us. So first up, we've got Professor Raghib Ali from Our Future Health. He is the CEO, Chief Investigator, Chief Medical Officer at Our Future Health in the UK, a clinical epidemiologist in the University of Cambridge, and consultant in acute medicine at Oxford University Hospitals. His talk title is Our Future Health, a world-leading resource for population health, genomics, and prevention research.
Thank you, and thank you to the organizers for inviting me. This is actually the first talk I'm giving, I think, to an international audience about Our Future Health. So I wanted to start by asking you, how many of you have heard of Our Future Health? Okay, so when I asked this question in the UK, it's about 90% now, and similar audiences. Here, it's less than half. So that's good, so hopefully you're going to learn something in the next 20 minutes. But in some ways, it's slightly surprising, and it's probably our own doing, because we haven't publicized what we've been doing at Our Future Health either in the academic literature or more widely, even though as of last week, I think we're now the largest consented cohort in the world with biobank bloods, as you'll hear shortly. And we have very big ambitions for this project and for this study. As you can see from the title there, we want it to be a world-leading resource for population health, genomics, and what was described as precision prevention research. So Patrick visited us last year and told us about PRECISE, and I was very excited when I heard about the project, because in many ways it's very similar to what we're trying to do in Our Future Health, and he's given a very good background to why we're doing Our Future Health already, so I can probably skip the first few slides in my talk. But you can see from the first slide, this is UK data. But it's a common feature across high and middle-income countries, really, across the world now, with aging populations, increasing levels of multimorbidity, common chronic diseases, and not just in older populations, we're increasingly seeing that even in working-age populations, and also ongoing and persistent inequalities, particularly by deprivation, but also with certain ethnic groups. In the UK, we have started to see some decreases in life expectancy, as well as plateauing of healthy life expectancy. Some of this is driven by the pandemic, of course, but some of it even preceded that, driven by increases in obesity and its consequences. As you can see from here, multimorbidity increases rapidly from the age of 50, and most people over the age of 50 in the UK have at least one common chronic condition. This is showing working-age populations, again, post-pandemic, very large increases in different diseases, but particularly mental health disorders, musculoskeletal disorders that are preventing people from going to work. So I'm mentioning all of these statistics as a way of explaining that these are huge public health challenges that we face as healthcare systems, not just in the UK, but across the world. As we heard, our current model of care is very much focused on treatment of patients with symptoms and signs of disease. Myself, I'm a consultant in acute medicine, which means that I work in A&E when I'm working in hospital and see patients with heart attacks, strokes, kidney failure, cancers that have already spread, etc. And of course, we know that that is too late, particularly to cure disease, but even for treatment of disease, it leads to much worse outcomes. And our health system and model of healthcare has very much been based on this treatment of late-stage disease. And we want to move towards earlier detection, earlier intervention, and prevention. Prevention, as we've known for many hundreds of years, is better than cure. But of course, in practice, demonstrating the evidence for that is not so straightforward, actually. And Our Future Health has been set up partly to enable us to look at the evidence for how prevention, particularly what we might describe as primordial or primary prevention, but we'll have the opportunity to look at all these types of prevention can be used as early as possible in a patient's or in a person's life. Historically, at least in the UK, we have mainly focused on moving the population distribution of risk factors to the left, and that was very appropriate. Things like high blood pressure, tobacco intake, salt intake, etc. But now with newer technologies, we have the opportunity to identify people at highest risk and treat them. It mentions on this slide that they can be treated medically, but of course they can be treated both medically and through non-pharmacological interventions. This slide is by no means exhaustive, but it lists some of the relatively new technologies which we can now use, some of which we've heard about already at the conference, in terms of being able to identify people at high risk through prediction of risk, early diagnosis and early detection of disease, and also early interventions. So this is what we're trying to move towards, this new model of care, precision public health, where we can ideally start from birth, but in the case of our future health, we're starting from adulthood, so from the age of 18, to identify these people as early as possible in the pre-symptomatic phase, and then intervene early, thereby increasing not just life expectancy, but particularly healthy life expectancy, so the number of years that people live in good health, which has really been static for many decades, actually, in the UK and also in other countries. So that was all by way of background. Our Future Health was launched through the Industrial Sciences Strategy, so it actually came out of the Life Sciences Industrial Strategy by Sir John Bell, who many of you will be familiar with. He was until recently, Regius Professor of Medicine at Oxford, but also UK Life Sciences champion. And it was designed as a program, a resource to help people live longer, healthier lives through better prevention, early detection, and improved treatment of disease. And it was set up from the outset as a public-private partnership because it has multiple aims and objectives, not just to improve health in the public health system and to help the NHS in the UK, but also to help the life sciences industry and to develop new diagnostics, to develop new drugs and new biomarkers and new interventions. So you can see from this list of partners that our Future Health really has this very unique collaboration across multiple stakeholders in the UK. Of course, the NHS is the most important in terms of our healthcare system. You heard this morning from Dame Sue Hill about the work that's been done in very impressive work that's been done in genomics, but across the UK, as many of you will know, we have the vast majority of our healthcare is delivered through the NHS, and that's obviously a key partner for all that we want to do in terms of our intervention studies and also prediction and screening programs through our Future Health. We're also working closely with the government and industry, so our initial funding came from government, but then that was more than matched by industry funding. We have many of our industry partners in the room here as well. We also work with a number of medical charities. You can see Cancer Research UK, British Heart Foundation, etc. And as I said, this was a very deliberate strategy from the beginning to ensure that all parts of the life sciences and health ecosystem in the UK are involved in the co-design, co-production, co-delivery of this program and can realize the full benefits from it. What most people have heard about our Future Health is it's a very ambitious aim in terms of the size of the cohort. So the first cohort study that I worked on was UK Biobank back in 20 years ago now. It was my first study as a very junior researcher, and at that time I thought it was amazing that we were going to have this study of 500,000 people, and it was amazing, and although it took a few years to get going, by 2010 we had recruited 500,000 people, and we have very much learnt from the experience of UK Biobank in terms of what we've done at our Future Health. There are some key kind of differences with UK Biobank, which many of you are familiar with. So it's not just the scale, but also the diversity. So we plan to recruit a much more diverse cohort, both in terms of deprivation and socioeconomic status, and also ethnicity and age. And I'll show you some of the data for our participants so far. But even more significantly in many ways, it's not just an observational cohort. We've set this study up for discovery and translational research studies, and we will be, when people consent to take part in the study, they consent to receive feedback about their risk of disease and also to take part in recontact studies, so clinical trials, biomarker validation studies, etc. And we also have set it up as a biobank, so as I said, we have over 1 million people's blood stored now, and it will be a resource, as I said, for the whole world. So in addition to the key kind of public health challenges we are seeking to address through our Future Health, here are some of the specific aims. So the first, as I said, is the resource for discovery and translational research for academia, for the NHS, for commercial researchers, the charity sector, and for the global community. Secondly, we want to be able to give people information about their personal disease risk for different diseases. We heard just now about some of the work that's already been done in Singapore. Some work has already been done through Genomics England and other projects in the UK as well, but nothing on the scale that we plan to do through our Future Health. And again, we're working closely with Sue and colleagues in the NHS to deliver this program in the coming years and decades. And thirdly, to go back to our participants and ask them to take part. So they've already consented to be recontacted to take part in biomarker validation studies, clinical trials. This could really transform the clinical trials landscape in the UK because we will have a cohort of millions of people who have already pre-consented with information about their health, about their genetic risk of disease, as well as other information that we're collecting to take part in clinical trials. I'll speak briefly about the opportunities in relation to polygenic risk scores. In practice, we will not be giving people their individual polygenic risk scores, but it will be done in combination with other risk factors, and starting with the diseases like type 2 diabetes, ischemic heart disease, where we have existing risk calculators, and we can add the genetic information. And that's particularly important for younger people who would be missed by current calculators, which are very much focused on age, appropriately so. But there are people, particularly from South Asian backgrounds and other ethnic groups who are at high risk, for example, of type 2 diabetes and ischemic heart disease at much younger ages, and were missed by our current early detection programs. For breast cancer, again, there are women who are at higher risk who are currently missed by screening programs. Again, we've heard about a study that's been done in Singapore. And we hope through our Future Health that we can establish the evidence base for these new approaches. Adding genetic information to existing risk calculators for diabetes, heart disease, breast cancer, in the future, other cancers as well, colorectal cancer, prostate cancer, et cetera, where screening programs don't exist. But there's lots of work to do here. We need to look at both the positives, in terms of how that can improve clinical utility, but also cost effectiveness, and also, of course, any harms that result from screening. So this is a long-term project, but it's certainly one of our important objectives. We are, though, also very cognizant of the fact that most risk factors for common chronic disease are not genetic, and at the individual level, certainly genetics is important, but at population level, of course, all of these other factors are more important in many ways, and so we do collect information about the environment, about lifestyle, about habits, etc. And we have a fairly comprehensive baseline self-assessment, similar to UK Biobank, which tries to collect as much information as possible about our participants, and then also through linking to NHS records, we can supplement that. Our Future Health is relatively light in terms of its baseline phenotyping. Of course, to recruit a cohort of 5 million people, it's very difficult to go into the kind of depth that we had with UK Biobank or even Precise. And so its unique features really are its size. And so we have kept the baseline phenotyping relatively light. So people fill in a questionnaire, give blood samples, have some physical measurements taken, blood pressure, point of care test currently for cholesterol, weight, height, waist circumference. But in the future, we can supplement that with a lot more data, particularly where data can be collected remotely through questionnaires, through wearables, etc. We can also link to additional databases with participant consent, to administrative databases, to census, etc. And also on our stored biosamples, we have the opportunity to do whole genome sequencing, proteomics, other omics, etc. But of course, that has an associated cost, particularly when you're looking at millions of participants. But the potential opportunities for phenotyping in the future are really very great. And thinking of dementia, for example, being able to track cognitive function across the life course, particularly at older ages, but that will be really something that hasn't been possible before. So the study it's a digital, predominantly a digital journey. You consent online after reading the information sheet. You fill in a questionnaire online, book an appointment online. But you do have to attend an appointment to give your blood and have those physical measurements taken. As I said, we are relatively new. We only launched in October 2022, so less than 2 years ago. Fortunately, we've had lots of positive press coverage, which has been helpful, although the majority of our participants have joined through our partnership with NHS DigiTrials, where we write to people to invite them to take part in the study. We have pioneered kind of an approach to make it as easy as possible for people to attend their appointments. So we have clinics in shopping centres, in supermarket car parks, and also in community pharmacy, something that hadn't been done previously. So we had learned from previous studies that shopping centres and mobile units were very effective. To that, we added community pharmacy. And with the first study, I think in the UK, certainly, but also globally, that's managed to show that community pharmacy can be used in this way to recruit very large numbers of participants and take blood from them. And more than half of our participants now have come through that route. Another key focus for us was to ensure that the study is as representative as possible, particularly in terms of ethnic diversity. The UK has become a very multi-ethnic country. Nearly 17% now of the country is of white background, but we also just want to make sure that people are not excluded for financial reasons, so we provide reimbursement for people's travel and time, and we've provided many more locations to ensure that a clinic is within a 5-mile radius of where they live. And you can see really very remarkable numbers of people that have decided to join the study already, something we've never seen before, I think, anywhere in the world. So nearly 1.8 million people now have consented to join the study. Of those, over 1.3 million people have submitted their questionnaire, their complete questionnaire. And as of last week, over 1 million people have given their blood samples. And within the next month, we'll have what we describe as a full participant, which is questionnaire and blood on over 1 million people, which I think, as I said, will make us the largest cohort of its type in the world. So although we have a long way to go, we've made good progress so far. And you can see roughly now we're seeing about 60-odd thousand people per month joining as full participants, so more than a person a minute. And we have, this is a little bit out of date now, but we have good coverage across the UK in terms of people joining. So the concentration is mainly based on population density that you can see there. But we have participants from every single part of the UK, which again has not been possible before, and from every age group and every ethnic group. This is the point I was making about the unprecedented kind of recruitment rate that we've managed to achieve, and at relatively low cost, so much lower cost than equivalent studies. In terms of the demographics of our participants, you can see a fairly good distribution across gender, age, ethnicity, and deprivation. We would like to do better. We want to make sure that we have adequate numbers from all people of all backgrounds and all ethnic and socioeconomic backgrounds particularly, so that they can also benefit from the research that we're doing. But we have made a good start. And as you can see, in terms of absolute numbers, it's already the largest cohort in the world of 18 to 40-year-olds, and it will be the largest cohort of non-European ancestry soon as well. This is a similar graph for the full participants, but I will skip through some of this. By ethnicity, we have noticed, as expected, and as we heard about this morning, about trust. And one of the biggest factors about people taking part in this type of research, and particularly people from less or more deprived populations in the UK and ethnic minorities, they are less trusting of these types of studies, and we have found that. So again, we have more work to do, but the distribution so far is better than previous studies, but it's not good enough, and our ultimate aim is to ensure that 500,000 of the 5 million are from ethnic minorities. So this just summarizes some of the unique aspects of the cohort, some of which I've mentioned already, but the opportunity to go back to participants to do recall by genotype, recall by phenotype, clinical trials, this multi-ethnic cohort, the very large cohort of young people, particularly important for mental health disorders, obesity, addiction, etc. And lots of these are huge public health challenges and haven't been possible before. Nearly all cohorts previously have been in the 40-plus age group. Also, lots of opportunities because the data set is so large for artificial intelligence and machine learning. I hope these slides will be shared later, so those who would like to look at some of these points in more detail will have time to do so. Just towards the end, I wanted to mention that the data from Our Future Health will be available to researchers all over the world. In fact, it's already available to researchers from academia and the NHS and small companies across the world. For large companies, you have to join as an industry partner to access the data. But you can now already access our data through our website. There's already quite a lot of data there, so nearly 1 million people's questionnaires, genetic data on over 300,000 people, and NHS-linked data on over 600,000 people. The UK is really the best place in the world to do these types of very large studies because of the single healthcare system. It provides an opportunity to link for every single participant to their health records, particularly for secondary care, diagnostic, and death data, but also, hopefully soon, for primary care data. And this really is the only place in the world you could do a study of this scale and this ethnic diversity. We already have quite a lot of researchers that have registered to take part, as you can see. We will have a lot more data soon. So by this time, or by the end of the year, we'll already have genetic data on over 500,000 and linked data on over 1 million people. And by this time next year, it'll be about 1.5 million for all of those categories of data. So a really very large and hopefully a very big opportunity for all of us to use this resource for discovery and translational research. And it will increase further in the coming years. So by 2027, we estimate we'll have data on 3 million people, including imputed data, physical measurements data, and newer data types. So I think this is my last slide. I mean, we have, as I said, very great ambitions for this program. It's something which provides opportunities that haven't been possible before. Ultimately, we want to make sure that it's used in a way that enables everyone to live longer, healthier lives. That's only going to be possible by the people in this room and outside of this room, researchers from healthcare, from academia, from industry, making use of the resource to ensure that we can realize those opportunities and those ambitions. So thank you.
Great, thanks for that really exciting project and amazing progress. So remember to save up your questions. If you've got questions, save them up and we'll do those after all the speakers. So we'll go into our next speaker. It's Dr. Rob Yang. He's a former pharmaceutical executive and current co-founder of CartaBio, championed tech-enabled decoding of human biology to advanced medicines. His title talk is Decoding Cellular Intelligence to Accelerate Drug Discovery. So over to Rob.
Okay, I'm going to adjust this a little bit. Thank you very much for the organizers for inviting me. I'm going to bring a different perspective or a diverse perspective. My background is computation and how do we use all the data, wonderful data we've collected over the years, and how do we translate that into drug discovery and biomarkers and into the clinics. So a little bit about me. I guess I was invited for my corporate pharma hat. But during the journey, I kind of switched out, and now I'm wearing the hat of entrepreneur. So for this talk, I'm going to share a little bit about past, how pharmas are using these data, how we're doing the computation, how we're translating it. But then I'm going to switch gears and talk about sort of the next present. I don't like to use the word future, but the next present, what type of data that we should be collecting, or at least my bias says that we should be collecting in order to accelerate. So spent about 13 years in pharma. Kind of, you know, I'm in Boston, so it's sort of like it's de facto to kind of go through the pharma. So Pfizer, AstraZeneca, Celgene, BMS, and J&J is where my last gig is, where I was leading a global team of data scientists and biomarker scientists. UK Biobank, FinGen. I think all of us and all of that cohorts are sort of what we have talked about as pharma to make investment because we believe in the data we're generating and we believe in the translatability. So this data or this slide I'm showing you here is that on the y-axis we're using different modalities, right? So quite often we're hearing multimodal modalities and how do we compute and create a multimodal model for that. On the x-axis are actually the resolutions that we're generating data from, and this is the aspect that I wanted to stress a little bit more for today's talk. We start off with tissue and organ just because technology-wise, that's where we were able to get the most out of it, right? So biotech and pharma, a large of what we do is depending on the technology that can generate data and compute data. So in the past decade or so, because of NGS and our ability to study genes and genetics, we're able to kind of get a lot of mileage out of our understanding of tissue and organ, and starting from a gene-centric framework for drug discovery. Now, you know, there's a movement on both ways, right? So down below, there's a single cell level component of it, right? So there's a huge movement to understand within the research community how we can understand not just on a tissue level, but also on the single cell level of phenotypes and so on and so forth. And then all the way towards the whole human part, there's the whole digital health and digital biomarkers side of things. So I just kind of want to kind of pave the landscape of biomarkers and the type of data that we've been thinking about. So my prior focus, now I'm switching over my pharma hat, right? So it's really based on bulk profiling, right? So, and then how do we actually use, you know, an average signal to create these biological associations where we can actually decode human biology and associate that with phenotype of interest. So, one of the drawbacks of working in pharma is that there's a lot of cool stuff that is not in the public domain. So this is just sort of the things that we have published, where we have used largely protein-based biomarkers to look for that progression, right? We have looked at using EMR, for example, to understand at the human disease sort of perspective, how do we actually stratify the future progression of certain disease in patients? We have used multimodal omics to understand, can we understand etiologies of certain diseases for drug targets? We have used imaging, right? So low-dose CT. Can we actually create AI models to understand and help clinicians to diagnose and match drugs? And certainly we have used large sort of pharmacodynamic cohorts where now this is multi-trial on the same drug, and we're trying to back calculate the drug MOA, the mode of action, and things of that nature. So all of these is sort of the present of what biomarkers, how we think about biomarkers in pharma, and how we're translating that into targets. And the big sort of magnifying glass I'm showing you here is that most of this, because of cost, because of convenience, we have sort of focused on that tissue and organ level. Now this is where I'm switching gears, right? But I do wanted to kind of talk about single cell type of analysis. Single cell so far have been sort of a newer field where the application and utility of it have been mostly on the research side of things, trying to understand, trying to create that dictionary of cell types and cell substates. But if we step back a little bit and think about us, human biology, right? So, me using my computing science background, what I think about is that there's layers of complications and complexity that associate with how each one of us is being made. At the bottom, we have the genetic codes, right? So these can be your programming language and your programming syntax, where now we're trying to understand genes and DNA, RNA, and proteins, individual drug targets. Above that, we actually have 37 trillion cells in our body, right? Each one of our cells is actually an autonomous driver. They are capable of making their own decisions and whatnot. And their makeups are largely the same. It's the same DNA that kind of codes for about 20 to 25,000 proteins and so on and so forth. But together, when they're put into certain contexts, known as cell type and cell states, they actually behave differently. And then one above that is how they're reacting and acting with each other to influencing each other, right? So what my entrepreneurial sort of journey I want to share with you is sort of like now we're focusing, and we have the technology now to focus on large cohort and generating large single cell type of data for large cohorts. Now we're trying to get at the app level. Now we're trying to get the operating system level of human biology. And the other side of looking at it is that in terms of the clinical success, we're actually seeing quite a bit of inflection point right now. There are questions about designing ADCs to target specific cell types. There are sort of bispecific programs that target specific cell states or immune states. So we're seeing that success in the clinics. And then the other part is that when we actually retrospectively analyze a lot of these MOAs of past drugs, we're actually finding out by serendipity these drugs actually work by targeting specific cells, right? So these are the sort of more back translation evidence for us to focus a little bit more on what I call as the cell-centric drug discovery. This is an actual example. This is a phase 1 drug. that targets a common target, or at least a target that multiple farmers are targeting. There is a preconceived notion of how this drug works, right? So there is an MOA that says this drug, after targeting this protein, should actually push the T cells to a specific state. But when you actually done single cell analyses on the actual human in phase 1 study, there's a couple of observations that actually shocked us or surprised us, and this actually leads to fundamentally why my current bias is that the cell state should actually be inserted into that whole drug discovery framework, right? So going from drug to target to cell state to actually the endpoint of diseases. So this particular drug actually was able to achieve a certain cell state that we later on through analysis found that was the more desirable cell state. However, it also has state 4 and state 5, which again, through later analysis, we show that not only are they not beneficial, not only are they not what we, let me rephrase that. They're not just neutral substates, right? They're actually counter beneficial. So the conclusion out of this example is that A, we don't know how our drugs work until we hit the clinic, until we look into the single cell state, and B, how do we actually do this more thoroughly, built into our large cohort data, so that from the get-go, from drug discovery, the beginning of the drug program, we're already thinking about these things. So the challenges of understanding cell states is that A, we don't have a cell reference, and B, we don't know which ones are pathogenic, and C, even if we know all of that, how do we actually manipulate, how do we actually rewire, how do we actually re-engineer a lot of these things for our own benefit. So one way to think about these building tools and solutions for is, again, using our ability to actually compute now, right? So you often hear about AI in drug discovery. There's different flavors for all of them. There's one flavor where now we're using foundation model, the concept of foundation model, where we pre-train using non-labeled single cell data to understand what the fundamental rules of parameters on how the cells are behaving, right? So the Mother Nature had already made up all the rules, and what we're trying to do is using AI to create and decode and reverse engineer some of those rules. And just like ChatGPT, right? We don't necessarily need labels for each one of these words to now do machine learning. We can pre-train using largely unlabeled, creating that transferable learning and parameters, so that we can later on create machine learning where now less data are needed. So, and then we also need a way to measure at scale. So, I think our cohort studies have largely kind of been built measuring genetics and circulating proteins. I think there is an aspect of it where we can now have the technology where that cost is low enough. We have one and others have others, where we can actually do this more routinely. So now single cell is no longer just a research level of a tool, but also a clinical stage level tool. And then at the top, the application of it is how do we actually translate all of this computation into drug targets, into our ability to match drugs to patients, and into understanding a lot of how the current drugs work. So going back to the case study, we've shown in this case, we built out a dictionary, and it's currently the world's largest T-cell substate dictionary, and we're able to kind of use this reference to explain what is that cell state 3 that we're targeting, and again, how are cell state 4 and 5 through across diseases, why are they actually pathogenic, and the next step is essentially that, hey, look, we can stratify patients based on their before drug cell state composition, and B, we can actually improve our current drugs by pushing a lot of these cell state 5 and 4s into what we really want. And then the next question is, okay, all right, how do we actually push? How do we actually push state 4 and 5 into state 3? And this is where, again, that AI simulation kind of comes in, right? So, so what we're able to show is that the foundation model that I told you about, that we pre-trained on 63 million cells, could actually capture a lot of these cell type specific wirings within their gene network. And we're using all of that information to create a simulated sort of knockout perturbation effect, right? So where we knock out computationally one or two at the same time and go through the permutations to rank whether the destination cell state is ending up to what we wanted to do. And certainly this is intractable to do in wet lab, but we can do 10, we can do 20 of these critical experiments. So this is, again, an example of where AI could actually help us ranking our hypotheses so that we can actually get to the right answer a little bit faster with higher probability. And then the question after that is, okay, we talk about T cells, we talk about CD8, CD4s, and we even talk about exhausted, non-exhausted T cells, but are we actually talking the same vocabulary in reality across different clinical trials, across different diseases? And the answer is, when we project everything onto the same latent space, onto this reference dictionary, we're able to see that different patients, different sub-diseases actually are driven by different T cell subsets. So this kind of gives you an understanding of whether, again, the same drug can be expanded into certain indications, and then the surprising things that come out of it is that when we actually cross-reference and project the in vitro assay where we're capturing our T cell states with the human T cells from clinical trials, we're able to see that the driver in vitro T cells that we use for drug discovery is not good enough, right? It only captures certain states that's not representative of what is actually driving the disease in the clinics. So this is huge because if you think about these things as your GPS, if your GPS is wrong from the get-go, it doesn't matter if you create a Lamborghini at the end, it's a beautiful molecule, right? But it's not going to the right place. And then later on in the clinics, that's when you actually find that's where the problem is. So as part of this. This discovery, we're partnering up with pharmas to create sort of cell state specific preclinical assay, right? So now, if you think about precision medicine, this is how we think about it. We're actually thinking about a convergence, right? So one is back translation, decoding human, understanding the cell type, cell states, that's driving certain diseases in the clinics. And then the forward translation is that we're doing perturbation, we're making changes in disease models, and then the neutral overlapping currency that we're comparing them with is cell state, right? So is certain drug in our specially designed assay really capturing the same cell state that will translate into clinical benefit efficacy. So that's sort of how we're able to co-create this precision medicine forward and back translation map. And then there's the biomarker clinical utility of it, where now the idea is if we can actually measure in every single patient, single cell level of data, and therefore using that to segment patients, then we have a better chance to explain where they are in their patient journey, and then match it with certain drugs that we understand the MOA of, right? So, so this is part where we're creating a few partnerships where we're piggybacking on existing biobank, and then we're creating another layer, another slice of the modality in saying that look, in addition to, at the bottom, our gene level understanding, now we have another tool, another set of weapons to understand the middle layer of cell type, as well as the top layer of the cell-cell interactions. So coming back out all the way, I think we're moving towards, from a drug discovery translational point of view, on how to use all these data is the following, right? So it's a cell state focusing, and then at the top you have your instruments, your computation, and your measurements. And then at the bottom, you would actually have your application, your clinical and preclinical sort of ways of driving and measuring a lot of these things. And you really need all four of these to have successful translational biomarkers that can also explain enough of the biology so that you can start drug program, right? So this is the biomarker to target connectivity here. I think this is my last slide, and in the last 20 seconds. So this is the landscape of who's the early movers in pharma, and that's doing this type of thing. We're working with Biogen, or we're working with BMS and Sanofi on T cells for their autoimmune and oncology programs. We're also working with Biogen to look at indication expansion and things of that nature. So I think we're coming, I think my sharing of the journey and my bias today is that there's different data types that's coming onto the scene, and my hope is that for people building future data cohorts and things of that nature, this could be an aspect for consideration. So with that, thank you very much.
More speaker in this session, and it's my pleasure to introduce Heidi Rehm. Heidi has a long history in data sharing and genomics and clinical genetics. I guess primarily she's the director of the Genomic Medicine Unit at the Center in the Center of Genomic Medicine, and Chief Genomics Officer at Massachusetts General Hospital, working to integrate genomics into medical practice. And our title of her talk is Advancing Genomic Medicine Through Global Data Sharing and Collaboration. And over to Heidi. And remember, we'll do questions afterwards, so that'll all be done through the app and after.
Great. It's a real pleasure to be here. This is the first trip I've made to Singapore, and it's a beautiful country, so I hope to come back someday. All right, so I'm going to talk about, as Thomas said, how we've been working to advance genomic medicine through a lot of different approaches. I'm going to talk about this in the context of rare disease, which is the main area that I focus on. So just to sort of set the stage for those of you who don't work in this space all the time, we've discovered close to 5,000 genes for rare disease, but still every time we run a case in the clinic, about over half of the time we can't find the answer. So we're clearly missing a lot of genes and variants and their causes for disease, and there's actually evidence for over 5,000 more genes yet to be discovered. So what are we doing wrong? How do we find all the rest of these genes? So we've put together a center over the last decade to really work on addressing the causes of rare disease. It's led by Anna O'Donnell-Luria, Mike Talkowski, and myself, and have just an amazing team at the Broad Institute where we focus on new methods, new approaches, analytical and technological, to try to identify novel causes of rare disease. So I'm going to give like a fairly high level overview of where we are today and the progress we've been making. One critical aspect of that is partnering all over the world with amazing scientists that recruit their own families and patients. And on the left side of this slide is many of the investigators we've worked with to amass thousands of samples, as well as working in partnership with patient advocacy groups that are recruiting their own patient populations. And then we have our own Rare Genomes Project, where we directly recruit patients into our program. So it's both a collaboration with samples from other investigators and our own direct recruitment. One of the critical parts of the work we do is making sure that everything we do can be disseminated and be accessible to other groups. So for example, we built a genomic analysis platform. This started when Daniel MacArthur and I started our center. But we continue to distribute this platform as a free open source platform that anyone can use. And there's actually now over 2,000 investigators around the world that have used this platform. Some have installed it locally on their own environments. Others use the main instance that we put on the Anvil platform that's funded by NHGRI in the US. And it allows us to incorporate structural variants, RNA-seq analysis. We're constantly adding new features. We have a node that hooks you up to the Matchmaker Exchange for collaboration. It's FSMA compliant in terms of its security, so it really is a great platform if you want something that's free. I encourage you to check it out. But now I'm going to turn and just talk about our own progress trying to solve these genetic disorders. And so what you see on this slide is in our hands, we normally are taking in samples that have already had clinical testing and have tested negative. Despite that, we're actually still able to solve about a third of them are positive over time in terms of our discovery. And then sadly, 45% are still negative, and then about 20% inconclusive. And that ranges from novel gene discoveries through our own work and our collaborators. We've discovered over 300 novel gene discoveries, but some of them are just identifying known genes and variants in them that have been missed through clinical testing. So I'm going to dive into the negative and conclusive buckets. So what are we missing, and how do we find the things that we're missing? So that we could be missing something because the actual sequencing data doesn't have the answer there. And so in a lot of cases, we start out with exome, and moving on to genome gives us other variants to look at. And in our hands, if you really scrutinize what required genome sequencing, and we just published this recently in the New England Journal of Medicine, in our hands, 8.3% added yield from genome analysis. And let's see if this pointer works. No. The very dark bars there are the things that are being added by genome analysis, mainly structural variants, other variants that are in poorly covered regions of exomes, as well as deep intronic variants, which are really being helped by tools like Splice AI that are pretty good at identifying cryptic splice sites and disruption of splice consensus sequences. Also, tandem repeat expansions, and then the most recent major neurodevelopmental gene discovery of RNU42, which was before we published this paper, but actually as a non-coding RNA gene adds a huge fraction, just that one gene, mainly one mutation. So that's where this added yield is coming from. This is an example of that paper that Nikki Whiffin's group recently published with hundreds of collaborators, because this variant is probably the single most common cause of neurodevelopmental disease. It's just amazing that it took us this long, and it's because it's in a non-coding RNA gene, and we just haven't done a good job at looking at the non-coding regions of the genome, both genes and other areas. We are also applying long-read sequencing approaches, both through ONT and PacBio. We've been making some discoveries, although I have to admit, a lot of them are things that we could have actually found from short-read sequencing. But there are some of these discoveries that are being aided by long-range sequencing. And I think as we build better population data sets from long-read, it will help allow this technology to yield more insight. We also now thinking about, all right, we may have the data there, but we're not interpreting it well. And so how can we do a better job of that? And in our hands, RNA-seq has been a really wonderful tool. This is just an example from a case of a patient with centronuclear myopathy. The actual specific gene, MTM1, was suspected by the physician, but it wasn't until we added trio genome sequencing to this family in RNA-seq that we could identify a major outlier in expression and then through splicing analysis, be able to see the skipping of an exon, and it turns out there was a line 1 element deep inside the intron, and thus finding was also mosaic in the mother. And so this case was quickly answered with just adding RNA-seq. Another more interesting case where we had a huge range of underexpressed and overexpressed genes. We could not, actually looking at all of them, find the variant that was that causal, then discovered that this case was a huge outlier in intronic retention. And we could see certain introns being included, and it turns out that this was also a small nuclear RNA gene that controls the U12-dependent minor spliceosome complex. And so this actually contributes to about 0.5% of introns across the genes in our genome. And so random particular exons being affected in terms of this. And that's why so many genes are being affected. Some showing underexpression, probably because the transcripts were rapidly degraded. Others showing overexpression because of the inclusion of the intron material, making it look like a higher signal. So this was a really interesting case, again, solved by RNA sequencing. In total, focusing particularly on muscle diseases where it's easier to get access to disease-relevant tissue, we've been able to add 14% yield with RNA-seq. This is work led by Vijay Ganesh. So that adds additional yield there from the RNA-seq looking overall. But I will say the most highest yield tool that we have is just relooking at the data over time. And this is just the reality of the constant churn of new knowledge, new gene discoveries, new variant discoveries being put out there, and being able to solve older cases. And so this is an area that we're working on because our team has been going back manually every single year, relooking at every case, and that's a lot of work. So we've been working through a collaboration with Daniel MacArthur's group and Microsoft on developing an automated process. Our automated interpretation pipeline that we used to call AIP is now called Telos. And we've been validating it both in Daniel's group with Zornitza Stark and her team in Australia, and also our data at the Broad Institute. And it's been quite high yield in terms of detecting already identified variation when we were validating it. And it performs better than some of the other tools like Exomizer and Lyrical. And now we've been focusing on novel identifications from this tool and finding new variants. We've been also adding additional features like better specificity for phenotype matching and so on. So this tool is continually evolving, both in Daniel's team and our team at the Broad. And one of the things that I'm now working to do, because we all are dealing with this, the fact that we need to reanalyze exomes and genomes. So we're working through a partnership with the Anvil platform to have a path to bring clinical laboratory data onto this pipeline and allow it to be subject to this automated reinterpretation and provide this as a service, and also allow this data to be immediately viewed for free in the Seeker platform. And then the physicians who would be facilitating their patients getting consented for the data to be available on this platform would then get emailed if a new finding was identified in their data set. So we're working to really stand up this pipeline so that any patient and any physician who consents them can bring this data on and have it subject to an automated pipeline. I think some clinical labs are really working to build this out, but most clinical data today gets looked at once and then sits there forever, or gets discarded. So this is something that we really need to think about, is getting that clinical to research pipeline instantiated. So that's, in a nutshell, some of the approaches that we've been taking. I think now switching to the inconclusive area where we've candidate variants or candidate genes, and thinking about how we support building the evidence for those genes and variants. And so a number of years ago, a group of us worked together to launch the Matchmaker Exchange. That was one of the first federated genomic data sharing platforms. We built that platform using the standards built by the Global Alliance that allowed federated sharing of data. So in this case, all of these nodes all have data, but it doesn't leave their hands. It's in their own countries, their own institutions. But through a common API, we're able to query across this network. It's been, I would argue, the single most valuable tool for rare disease gene discovery of anything we use. And it's really built on data sharing. And the idea is you have a candidate gene, you enter it in the system, and you query the other data sets. And if there's a match, somebody else had entered the same gene, you get a hit, and then you collaborate and you publish your paper. From our site alone, we've submitted over 1,500 genes, received 500,000 electronic match requests that led to 13,000 matches in our Seeker database. The other nodes are doing similar things. Gene Matcher is one of the most widely used tools because anyone can set up an account and use Gene Matcher. So to date, there's over 15,000 candidates in this system. It's led to thousands of gene discoveries. So this has just been an incredibly powerful tool that's been based on global community data sharing. So that's great. Almost every discovery we've made in the last decade has benefited from this platform. However, sometimes we put in our candidate and we don't get a hit. Then what do you do, right? Or your VUS, it's a VUS and a known gene. This platform is not made for known genes. It's just for novel gene discoveries. And so what do you do with your random VUS that plagues you? So we've been working on the next phase of this federated data sharing, and that is what we call one-sided matching. So at the top is the two-sided matching. Two groups have the same candidate, and you just bring them together. It's pretty simple, actually. But if you don't get a hit, and you just want to query all of the world's genomic data, so does anyone have this variant in their data set? And if so, tell me the phenotype of that patient. Wouldn't that be brilliant? If every variant you find in your lab, you could just query, find it, and get the phenotype. So that's what we're trying to do. You put your candidate in, and you match to the world's data. So there's actually some systems that have already launched doing that. Variant Matcher, the same group from Nara Sabrera and others at Hopkins that have launched Variant Matcher, similar to Gene Matcher. But this is me. Anyone can set up an account. This is me entering a patient with the phenotype of my patient, putting the variant in, and then getting back a list of 8 cases that matched and their phenotypes. And what I quickly saw was the phenotypes didn't match mine. TAC4 were healthy parents of probands. So I quickly ruled this variant out and moved on. That was incredibly helpful. But that's one data set. Do I have to query one by one 1,000 databases to do this? Well, that would be annoying. So we're trying to now, through a collaboration with the Global Alliance, and additional standards that have been built through now the Beacon version 2 API to start connecting genomic variant data stores all around the world where we can get access to the existence of variation even though no one has identified it as a candidate anywhere, and then be able to facilitate access to phenotype. And so the first 2 nodes that have just been launched are between Variant Matcher and the Franklin commercial platform. And this has enabled now cross sharing between those 2 platforms. But we have many other groups that are joining us monthly as we work on implementing this federated platform. And this is just a screenshot from the Variant Matcher platform where you can see when you enter your query into Variant Matcher at the bottom, it automatically says defaults to searching Franklin. Likewise, when you're in the Franklin database and you're searching your variant there at the top or another one at the bottom, you automatically are getting data from Variant Matcher. And that variant could say, it doesn't appear there, or it could say, yes, the variant was found 726 times, 144 are homozygous. That right there is enough to say, I don't need to go over to Variant Matcher and search. This variant's too common to be a rare disease variant. But if you wanted to search, maybe it came up once or twice, that looks more intriguing. You could click on that URL link. It would plop you into Variant Matcher, you log in and get the phenotype. So this is sort of how we're developing this, is seeing the aggregate allele frequency data immediately present, but then a link to get you in to log in and get the phenotype data. So this, these are some of the stats since this has been live. Franklin is actually, it's a huge platform. There's about 250,000 queries happening every month through that system. already been thousands of variants matched. And this is just the first 2 nodes. We're now expanding this massively. We're hooking our seeker database up. Many other groups are working to launch. So this is just a glimpse of some of the kinds of global data sharing that I think are really critical to advance our field and understand the annoying VUSs that we keep finding. So now I'm going to talk about one other approach to trying to share data to advance our interpretation of variation. So recently we launched Nomad version 4. And what you can see in these first 2 bars is we actually ended up adding a lot of European data because we added all of the UK Biobank exome data that we had access to. But what you see on the right side is a look at how many variants in Nomad, version 2 versus version 4, had an allele frequency over 0.1%, and separating by European and non-European data. And what you can see is despite a massive increase in European data from V2 to V4, almost no increase in the number of variants over an allele frequency of 0.1%. So it versus just a minor increase up to about 169,000 non-European samples in Nomad, we actually had a pretty substantive increase in the common variation that we were finding. So it tells us that we have largely saturated our understanding of common variation in Europeans. We have not begun to saturate our understanding of common variation in other populations, non-European. So it's really critical that we share data from lots of distinct populations, because this will allow us to filter out all of the common variants so we can focus in on the rare variants that are maybe causal for disease. So the challenge for us in building the Nomad database is there's lots of data sets that we don't have access to and can't physically bring to the Broad Institute where we build Nomad. So in addition to including the All of Us data set, which is an intentionally diverse data set that we'll be pushing out as soon as the 400,015 samples are out, we're right now focusing on what we call federated Nomad. And we've, in particular in Singapore, been working with Nicholas and Maxime, who've been awesome to work with in terms of getting both a Singapore data set represented in Nomad, but also many other countries that we're working with closely right now. And the idea is we are teaching them how to use the methods that we do to process Nomad data for them to process all of their data sets themselves, and then they share the aggregate allele frequencies with us, which we combine into the main Nomad data set, so that anyone using Nomad will have access to all of the country's data in Nomad. So that, we think, will really advance population allele frequency data. And I'll say this project is also being done in collaboration with the Global Alliance, particularly through the National Initiatives Forum, where we're bringing all these countries together. And I do want to just, in the last 10 seconds, is mention a little bit more about the Global Alliance. If you're not already involved, I encourage you to get involved. We have work streams that are really developing standards to enable data sharing, dealing with many aspects of the kinds of data sets and formats and file types and everything else, knowledge standards. We work with driver projects that tell us what's needed for data sharing and then develop the standards to support that. This is the National Initiatives Forum with lots of countries around the world with national programs that are participating in this effort, including the Singapore group here. And we've been really working, and this is work led by Catherine North as our fearless leader, to develop a toolkit for all the different aspects of things that every country needs to really support genomic medicine and trying to build the standards. All the little stars are already existing standards in the Global Alliance to support all the things that we need to do. So come back to this side of the world in about a month in Australia and come join our Global Alliance meeting if you're not already involved. With that, I have run out of time, so I'm going to have to skip the very last sort of clinical portion of my talk as the Chief Genomics Officer, but I'm happy to answer questions at the very end in our panel session. So I just want to thank a wonderful team that I get to work with on all these different projects, and most importantly, the whole global community that's just done an amazing job of working together to share data and advance genetics. Thank you very much.
well, I'll invite our speakers back up to the podium. So we're going to be using the pigeonhole.at app for doing questions during our discussion session, coming up in 30 seconds. So you can enter your questions. We already have some questions that are already there, so please enter your questions and vote for them as well, and we'll choose the base the discussion on the most voted questions. All right, okay. So let's get the discussion session started. I mean, we had 3 very different talks from very different aspects, and I think we've already started to receive some questions. So maybe we'll start working our way through some of those. So there has already been quite a few questions from the Our Future Health talk, and one of the obvious sort of questions is there a link between the Our Future Health and UK Biobank, and maybe to enable sort of some of the incorporation of the longitudinal data set and information from UK Biobank, and maybe the ability to recontact as well?
Thanks. So there's not a formal link, although we collaborate closely with UK Biobank and other initiatives in the UK. We haven't specifically targeted people in UK Biobank to join Our Future Health. In practice, the program is open to all UK residents, and so of the 500,000 participants in UK Biobank, a proportion, of course, will join Our Future Health. We haven't yet done that linkage, but we will do so in the future with participant consent to make sure that we can identify these people and have that additional, 15 odd years of retrospective data, as well as the ongoing follow-up through Our Future Health. In the UK, there are a number of large cohorts. So the Million Women Study is another one, 1.3 million women without genetic data. Again, some of those will be participants, and various other cohorts. So eventually, the aim is to try to consolidate all of the information from the various cohorts, so that we can identify people that have got multiple sources of data, and to make it easier for researchers to identify those people and their data. That's certainly part of the intention.
All right, thanks. It looks like for Heidi, there's a couple of questions that are being upvoted, and we can maybe try and do the first one. So, using the automated pipeline, how do you make sure there are no false positives? And that's the endless kind of question that everyone wants to know.
So the simple answer is that we don't actually make sure there's no false positives, in the sense that anything you find, you have to review and make a professional judgment that that's causal or not. The real goal here is, can we pull out the case that has something that's worthy of looking at carefully? And so that's our main goal. Over time, we're making it more specific, and I think to minimize the amount of work we have to do to review things, we're keeping the specificity fairly high. But that doesn't mean there'll be no false positives. This is not meant to be a direct return of a result to a patient. So, there's a balance there, and you can actually titrate it to how sensitive versus specific you want it to be, how close the phenotype needs to match, for example, between the gene and your patient's phenotype, and that might be dependent on how much in terms of resources you're willing to spend looking at the data. So, we're titrating it to be fairly specific, compromising some sensitivities so that it's a low resource sort of effort, but you can titrate it in different ways.
Yeah, I guess it's really about discovery initially, and
Yes.
then follow-up validation really for the checking.
Exactly.
All right, so let's go back to Our Future Health, which has got another popular question. Very impressive effort with Our Future Health. How does the NHS incorporate data from private health care institutes in the UK?
Thank you. So, as those of you from the UK will know, but for our international colleagues, there's a very small, actually, sector in the UK which is private compared to other countries. So more than 90% of our health is delivered through the NHS, and even higher than that for some diseases. So in general, we don't have data from private healthcare interactions in the NHS databases, but in practice, it's not a major limitation. Even those people who do see private providers, it's often following a diagnosis that's made within the NHS, and then they go to a private provider, for example, to have an operation done for which otherwise they'd be waiting a long time on an NHS waiting list. So based on previous studies like UK Biobank, Million study, etc., that have linked to NHS databases, it doesn't seem to have been a major issue, and so we don't anticipate that being an issue for our future health either.
All right, great. So let's switch to a question that we received about the Carta Bio talk. So, do cell state models incorporate the crosstalk between gene expression and proteomics? How is the bioassay efficiency of the cell state models measured against conventional bioprofiling? It's quite a lot there.
Yeah, I think for the first question, we see gene expression and proteomics as different measures of cell state. Our model currently translates from gene expression and infer protein and vice versa as well. And that way is just sort of like, you know, it's the multimodal translation type of thing that, you know, again, I go back to ChatGPT, right? So from image to text, text to image, that type of thing. But altogether, they all represent what we define as a cell state. So the crosstalk between protein and gene expression are not currently our focus. The second question, how's the bioassay efficacy? Yeah, this is a really good question. We don't know. We think the conventional bioprofiling is measuring the key markers that represent certain cell state. We kind of go one up on that by measuring more things that defines cell state. We actually don't know which one is better when it comes to pushing the drug efficacy out the door, but we think measuring more have a better success rate.
Great, thanks. All right, so let's go back to Heidi. So I guess you mentioned lots of different projects and initiatives, and one clarification is the difference between Variant Matcher and ClinVar.
So ClinVar has been an enormously successful data sharing project, but it's only sharing the interpretation of a variant, classification against pathogenicity, for a variant you've interpreted in a case. And sometimes the evidence is not there. A lab may submit and said it's likely pathogenic, and then there's no evidence for why. You have to reach out to the lab and say, did you see it in a patient? What was their phenotype? And you can ask more questions, but it's not right there. Whereas Variant Matcher is saying, we're going to open up our raw data, our entire set of variants found in cases, to anyone who wants to peruse them, even if we've never interpreted any of those variants. And so it's really looking for a variant from a data set, and then once you find that variant, asking what's the phenotype that's recorded with that case. So it's really looking at raw individual level data in databases like Variant Matcher versus ClinVar are interpreted variants that a lab has made a classification for.
Great. So here's a question that maybe, well, all of you could possibly comment if you've got a view on. This is the emphasis on partnerships with various public funded programs and industry, and this was certainly touched on in the Our Future Health talk. So how do we balance the inherent divergence of profit-based incentives and public health needs for a win-win? Who wants to go on that one?
Happy to start off on this. So Our Future Health, as I said, has been set up specifically as a public-private partnership, with the majority of funding actually coming from the private sector. And it's true that one of the issues which has come up from the beginning, and particularly as we've started recruitment, is participant concern and other stakeholders' concern about the potential for commercial companies to make profit from the project eventually through the discoveries that may be made. But in practice, both in terms of the general public and our participants, when we've explained to them the potential benefits that arise from this, and it's become, I think, easier to explain after the pandemic when people saw the collaboration, for example, between Oxford University and AstraZeneca to develop the COVID vaccine, but also, you know, other drugs that people use. Many of them have come about through collaboration between academia and industry, and many trials are done with the NHS. So at least in the UK context, it's easier to explain how working with industry leads to direct benefits, both in terms of new tests and new treatments for participants. And for me personally, as an NHS doctor, again, it's easier for me to make the case as to how that benefits patients and people in general. And I think over 80% of our participants and the public see that as a positive thing. So we are already seeing that to an extent in our Future Health, that people are beginning to see that this is a win-win. And certainly, if we hadn't had the funding from industry, it would have been much more difficult to get a program like our Future Health off the ground. So we have seen that benefit, hopefully, for everybody, and will continue to do so in the future.
Yeah, I'll also add, if you look at the ClinVar database, the largest data submissions are from commercial entities. And I think it's an example of defining a pre-competitive data sharing environment that's in the best interest of both private and public entities. And so there's a lot of different ways that we can work together to kind of define what we may do pre-competitive. And then those companies can still compete for the clinical genetic testing services they offer on price, on others, customer service, other things. But the actual data sharing in advance, we've really carved out as something that is pre-competitive. So that's one example. There are other examples. We've tools like Splice AI is a commercially licensable product for industry, but we've worked with Illumina to now define labs that submit to ClinVar and publicly share their data now get free access to Splice AI because they've shown goodwill to share their data. So there's ways we can actually incentivize and delineate sort of data sharing approaches to help companies sort of both keep their proprietary products and commercialize them, but also benefit those companies willing to share their data in ClinVar and other ways. Also, the UK Biobank project that we got all that data into Nomad, that was a pharma partnership that paid for all of the UK Biobank exome data to be reprocessed, all paid for by industry, but then it allowed us to put all that data in Nomad. So I think there's so many different creative ways that we can work together in a pre-competitive space that benefits both industry and all of us, working in this field.
Yeah, I just want to add on to that. The pre-competitiveness is what farmers make decisions on, right? So, a peek inside the war room when we decide on whether to invest into these cohorts is does this have values that will help the public and the patients in terms of pushing out drugs? And B, is this something that we can create certain data types that we think are useful? So for example, the UK Biobank proteomics data are largely paid by industry partners for that reason. And later on, that's all going to be shared to the public.
All right, thanks. There's a question for Rob here. Let me just bring it up. So here we go. So very interesting, Rob. How concerned are you about model collapse in the increasingly generative models that we trained on model-derived data?
Model collapse. Oh God. So we're concerned about the model being biased, right? So if you think about sampling, the whole premise is that we sample enough of the universe that the model knows what states are important. We haven't hit, in terms of computational limitation, we haven't hit the collapse stage yet. We're still on the up. But the bigger issue with generative model is that there is an intrinsic bias in terms of what we pre-train on, and that's the part that it's a little bit of unknown, right? So we don't know how to assess that.
All right. Just bring one up. So this one is for Heidi, and this is really looking at this issue about advancing variant interpretation. So what do you think is the most important thing we should do as a community to advance variant interpretation?
So, I mean, at the end of the day, it's really getting access to the evidence that's going to both inform variants, but allow us to scale it. We have billions of variants in our genomes, And there's not enough workforce to manually interpret one by one and search PubMed and every resource and email the people that submit to ClinVar. So if we're really going to advance this field, we need to share the underlying evidence in scalable and standardized ways. That's why I emphasize working with Global Alliance, where we're really trying to standardize how we share data and how to get access to that data through APIs and other methods. And that could be individual level data, that can be functional data, the results of scalable MAVE assays for variant function, as well as in silico algorithms, all the different evidence types that we're going to need to be able to use, making that data accessible as standardized structured evidence will also allow it to be computed on through machine learning approaches. Right now, we can't take advantage of machine learning that well because the underlying evidence isn't accessible enough in a lot of cases. So I think this is what we all have to think about is how do we each make our own data sets, whatever they may be, accessible and standardized in terms of data sharing. And that would, I would argue, will be the single most important thing to advance this field.
Yeah. All right. So let's see. A question that I had actually. I mean, we talked mentioned several times about different types of assays, particularly genetic assays. You know, there's a whole variety of different options at the moment. And we heard from Sue Hill this morning. You know, the targeted sequencing, exome sequencing, whole genome sequencing, long reads, short reads. How do you integrate or choose what's the most appropriate assay or you know, of those selection of options basically, and how do you integrate the knowledge that you can get, say, from long reads versus short reads? Anyone want to comment on that?
So, I mean, you know, I think the answer is we need them all, but I think by testing out these different methodologies, you can start to see what the added yield of each one is. And that's why we, you know, published the study looking at genome versus exome and looking at the yield of RNA-seq. And right now, I will say that RNA-seq is probably being observed by many groups as one of the highest, well, I should say reanalysis is actually the highest yield additional tool, but beyond that, for the technology side, I would argue that RNA-seq has shown us the largest incremental advances. And so you can say, well, we need to start doing that, but that's not to say that long read doesn't also give discoveries. And so I think the question becomes, how can we make each of these additional tools not cost so much money and done one at a time? Like, what ways can we integrate these tools all together? And right now, they're largely separate approaches that we're taking, which adds incremental costs. But I think over time, we'll start to really bring some of these technologies together as cohesive, you know, comprehensive approaches. But it's still kind of one new technology after the other.
Yeah.
From sort of the data modeler point of view, I used to think the same way, we need them all, the more data, the better. But I think over the years, we kind of through discovery, we're trying to kind of look at it from a drug discovery output point of view, right? So RNA-seq actually gives us a great amount of mileage, just because we are pretty good at computing and incorporating and interpreting now. The long reads is a little bit newer. We're looking at that for splicing and things of that nature. Whether that has a lot of drug discovery ROI, that remains to be seen. So I think you know, there's user-derived decisions on what data modality to generate, and then there's the academic discovery aspect.
I mean, just to add briefly, obviously for our future health, one of the key considerations for such a large cohort is cost. And so at baseline, we're doing, as I mentioned, genotyping and imputation. In the future, we would plan to select on the basis of certain characteristics subsamples out of the 5 million to do additional either whole genome sequencing, long read, short read, methylation, RNA-seq, et cetera, depending on the research question. But the second consideration, because we do want to translate discoveries from studies like our future health into clinical practice, where it can be used within, you know, in the UK and the NHS. And again, cost will be a key part of that. And if you can do a genotype for £10, that's a reasonable cost to start integrating into clinical practice. When you're talking about hundreds or even thousands of pounds for certain tests, it's much more difficult to see how that can be implemented at scale. So these are, cost is a key consideration for translating discoveries, as this whole conference is based on, from cohorts into the clinic.
All right, I'd like to thank our speakers for the session, and thank you for sending lots of interesting questions.